Chapter 7, Section 1, DNA binding proteins. Now, we have to keep in mind when we're talking about genomes in any proteins or any, nucleic acid, protein, polypeptide, whatever complexes, when they're going to be interacting with the chromosome, they're going to be interacting not so much with specific genes all the time, but with a collection of genes. Turns out that the bacterial chromosomes, archaea chromosomes, are quite a bit different than ours. Our chromosomes, our genes have the whole intron-exon things. Introns, the intervening sequence, the exons, the expressed portions of genes. Majority of our chromosome is the introns. Prokaryote chromosomes do not have these intervening or the intron sequences. Where our genes are responsible for the production of certain proteins, and whether these proteins all have to come together in one complex, like a cytochrome complex, those genes can be found spread across numerous chromosomes. Prokaryotes, since they only have one chromosome, all the genes are going to be on that one chromosome. And we find that the chromosomes are going to be, grouped together. I'm sorry, not the chromosomes, the genes on the chromosomes. Many times they're going to be joined together, grouped together in what is referred to as an operon, which kind of makes sense. If you have a new cell ability that needs to be, turned on due to an external stimulus, this new ability is based on the presence and activity of a handful of enzymes and new structural proteins that need to all be expressed at the same time. Why not join them all together in a series, A, B, C, D, E? A through E genes are all joined together in close proximity so that when A gets expressed, so does E and everything in between. This operon can then be under control of only one promoter region. Hopefully you remember promoters from Intro Cell Cell Biology Genetics, that a promoter is a sequence on a chromosome upstream being just before a gene, or here an operon, that is an area where proteins will bind that will recruit. Okay, that's what a promoter does. It recruits in transcription factors, which will then recruit polymerase to come and transcribe that gene, or here, transcribe this operon. We find that these operons, the expression of these, with their promoter regions, are going to be influenced by what are called regulatory proteins. Okay, regulatory proteins are going to turn transcription on and off for a gene or for an operon. They can bind at or near a promoter region, and they themselves bring in transcription factors, which will then bring in polymerase, transcription on. Or the regulatory proteins can bind and mask the promoter region, which means no transcription factors will be able to bind, which means no recruitment of RNA polymerase. Now it's interesting, proteins and nucleic acids interact all the time, okay? They are polar, they'll have some polar regions in proteins and the polypeptides, and, nucleic acids polar, so they will interact very well with each other, but it's non-specifically. What we're going to be talking about in this chapter are what are referred to as DNA binding proteins. Okay, this is a bit different than these proteins here, because these DNA binding proteins, due to their amino acid sequence, they are going to bind to specific sequences, GATC sequences, within the DNA chromosome. They are going to bind, and it's going to be due to the side chains, the R groups of individual amino acids in these DNA binding proteins that are interacting with the sugar phosphate backbone as well as the nitrogenous rings. If you think back to what you learned about the structure of the double helix, you hopefully remember there is a major and a minor groove due to the double helix structure. These DNA binding proteins are going to interact, and they're going to situate themselves within the major groove. So if you step back, and we're looking at this big picture right now, throughout this chapter, we'll look at specific examples. Big picture. Some external signal triggers a signal cascade of some sort inside the cell. Activators and repressors will either be turned on, turned off. If a repressor has been turned on, remember it's binding, masking the promoter, which means no transcription factors. No transcription factors, no RNA polymerase, no transcription. Activator, if this gets activated, not the repressor, the activator is going to bind, and then it's going to recruit in transcription factors to the promoter. These other proteins will bind here. Remember, notice I keep referring to these as transcription factors. Transcription factors, when they bind, first one binds, recruits the second. The first and the second bound recruits the third. 1, 2, 3 bound recruits the fourth, which then all four of them together then will recruit in RNA polymerase. So the activator just makes it so that the transcription factors have a greater affinity for the promoter region. The activator may also kick off any repressor that might be bound here. So, however it gets there, RNA polymerase is now here. It's now positioned, and it starts just at the end of the promoter here, which shows +1. +1 means this is the first DNA nucleotide of the gene or the operon that needs to be expressed. -10, -35, this is the upstream 10th nucleotide, upstream 35th nucleotide. -10, -35, we're going to see in this chapter, there are specific DNA sequences here that will recruit in different binding proteins to this sequence. That's 10, 35 nucleotides upstream. Downstream, which means into the gene itself, you'll find different, ribosome binding sites, structural gene, the actual gene itself, and then a terminator region. Transcription takes place. You now have messenger RNA. Notice there's no pre-mRNA here. There's no talk of introns, exons, spliceosomes, any of that. No. Here's a level of control. Who can bind here? Who can recruit in RNA polymerase? Another level of control. These signals could turn on, silencing RNAs and ribose switches. Silencing RNAs, ribose switches can bind to the upstream sequence of the messenger RNA. Upstream before the gene itself. They can bind and aid in positioning of the initiation factor for the ribosome binding for translation to occur, or they can interfere. If they're interfering, means that the ribosome, the small subunit, can't get there. Small subunit can't get there along with the initiation factor that was it formulated methionine, F-met tRNA, then the large subunit can't get there. No translation will occur. Given enough time of this interfering, enzymes will come through and just chop this up. Chop, 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 chop. That messenger RNA gone. Or the ribose switch, this RNA can bind and aid in recruitment, aid in initiation of translation. Many different things. Ultimately, you get a polypeptide, you get all the polypeptides together that need to be there, you get mature proteins. We find that when you look at the nucleotide sequences, where many of these proteins are going to be interacting, we find what are referred to as inverted repeats. Okay? Inverted repeats means basically the sequence of the DNA nucleotides. Well, further along, you're going to get the mirror image. If it's GATC here, GATC, then it's going to be CGA or TA somewhere else. Few hundred or a few dozen, a few hundred nucleotides away. This is what aids, or I'm sorry, not a few dozen, a few hundred, maybe a dozen or more nucleotides away will be separating these. We also see that DNA binds what are referred to as homodimers. These proteins are homodimers. Means that it's two identical peptides. Dimer means two. That protein, the mature protein, is two polypeptides. Okay? Got to remember, primary, secondary, tertiary, quaternary. So with the quaternary structure is two polypeptides. Okay? Two polypeptides that are the same exact polypeptide. Two copies, as you see right here. Two copies that will bind, interact, they'll insert into the major groove, minor groove, see how tight there it is, major groove, wider apart groove. The inverted repeats, notice TG, TG, GT. Hey, look, TG, TG, TTT. It's on the opposite and going in the opposite direction. AA, TT, GT, TGTT, AA, same. Inverted repeats. They are spaced far enough apart that the two halves of this binding protein, which is a dimer, can be positioned. One half binds this part of the repeat, the other half binds that part of the repeat. This allows this protein to be positioned where it needs to be in relation to the genes on this chromosome. We see that the overall structure of these DNA binding proteins, not only are a lot of them dimers, but you're going to see this helix turn helix motif. Two alpha helices joined together by a turn. So you're going to have the alpha helix, then a turn, and then an alpha helix. First one's a recognition helix, the second one's the stabilizing. First one binds to a specific DNA sequence, AGTC sequence. The second one stabilizes, interacts, helps keep it in position so it doesn't slide and fall off. There are numerous helix turn helix binding proteins that are out there that we have studied. E. coli is quite a few of them that we're studying. The big one that we'll talk about here in this chapter, the Lac protein, or the Lac repressor, binds to the promoter region of what is referred to as the Lac Operon, the set of genes that need to be expressed by E. coli in the presence of the sugar lactose in the environment. Under normal conditions, E. coli doesn't get exposed to lactose, so it's not going to express the transport protein to get the sugar in or the enzymes required to prepare lactose to enter into the glycolytic pathway. So here's this alpha helix, that's what this cylinder is supposed to represent, a turn, and then another alpha helix. So you can see it right here in the space filling model. Red, blue. Blue is the nitrogenous bases. Red is the sugar phosphate backbone. You can see here minor groove, major groove, and it allows for it to insert. One is for specific sequence, the other is for stabilization. Other DNA binding proteins that you'll see mentioned in the textbook or in the primary literature are going to be zinc fingers and leucine zippers. Zinc fingers, this one's going to be mostly eukaryotic. Has zinc finger. It is a protein, a DNA binding protein that is atoms of zinc that allow for binding. Zinc to the polypeptide with the zinc to the DNA double helix. And then there's also what is referred to, like I said, the leucine zipper. Uses spaced leucine residues. Remember, leucine's an amino acid. These have specific DNA sequences they will bind to, and then the leucine zipper, based on its name, acts as a zipper and helps in the unwinding of the double helix, opening it up so that the, not a replication bubble, a transcription bubble can occur.